Hello friends, now in this video we discuss HDL metabolism and this is nothing but it's a reverse cholesterol transport. The process is called reverse cholesterol transport. Because HDL is having the responsibility of extracting the surplus cholesterol from extrahepatic tissues and deliver it to liver, from where it is excreted out in the form of bile acid. So that is why it is called reverse cholesterol transport. The cholesterol is taken out from the extrahepatic tissues and it is delivered to liver. We'll see how this process is actually taking place. You will be understanding that one enzyme called LCAT, Lecithin Cholesterol Acyl Transferase enzyme, play a very important role in the process of reverse cholesterol transport. Liver as well as GIT, they produce nascent HDL, which is discoidal HDL. This discoidal HDL is having a shell which is having phospholipid, abundant phospholipid, and some free cholesterol. The shell is made up of phospholipid. This is shell made up of phospholipid and free cholesterol. This is free cholesterol, unesterified cholesterol, free cholesterol. So these are two components of the shell of this discoidal HDL. The core section is having minor quantity of cholesterol ester. And the nature of apoproteins which are there on HDL are ApoE1, ApoC and ApoE. This kind of discoidal HDL, once it comes to circulation, it activates one enzyme which is plasma enzyme, LCAT, that is lecithin cholesterol acyl transferase. This enzyme is plasma enzyme and this is activated by ApoE1 of the HDL particle. Now once LCAT is activated, it gets attached with the HDL particle and does its role. The role of LCAT is as follows. It converts the free cholesterol to cholesterol ester. And lecithin is the donor of fatty acid which is attached to the third hydroxyl of the cholesterol. Lecithin is a phospholipid and it's having the PUFA at the second carbon position, and that fatty acid, second carbon fatty acid, is being transferred to cholesterol as to convert it to cholesterol ester, and lecithin is in turn converted to lysolecithin, and hence the name of this particular enzyme is like this, from the lecithin to cholesterol fatty acid transfer. This kind of action where LCAT is converting cholesterol to cholesterol ester is actually taking place in the shell of the HDL particle, where one of the free cholesterol is modified, it's converted to cholesterol ester by addition of the fatty acid to the third hydroxyl, and this cholesterol ester, because it's a non-polar, it moves into the core section. So once LCAT is acting, one of the peripheral cholesterol is modified to cholesterol ester which moves into the core. Now this creates a space in the shell of the HDL particle which is called lacuna. So this kind of lacuna is produced when LCAT is acting on the HDL particle. This lacuna is the ideal place where extrahepatic cholesterol, this is extrahepatic cell which is having excess cholesterol in the pool. This free cholesterol, excess cholesterol from the extrahepatic cell is moved to the lacuna. And you should know there are transporters at extrahepatic cell which transport the free cholesterol from the extrahepatic cell to lacuna of the HDL particle. So this free cholesterol moves into this lacuna, and this new incoming cholesterol also is meeting the same fate, and by action of LCAT it is again modified to cholesterol ester when one fatty acid tail is attached from lecithin, and this cholesterol ester again moves to the core, recreating the lacuna. So some more cholesterol moves in to this lacuna, and that is again modified to cholesterol ester and moves within the core section. When repeated action of LCAT is seen, more and more of the cholesterol ester is accommodated into the core section of the HDL particle, and this becomes a spherical particle which is called HDL3. HDL3 is having cholesterol ester in the core, more cholesterol ester compared to discoidal HDL, because LCAT action converted peripheral cholesterol to cholesterol ester and that moved into the core section. So it will have more of the cholesterol ester in the core, and that is why its shape is becoming spherical. The shell is having same fashion, the phospholipid, the free cholesterol, and of course the lacuna, because whatever cholesterol is coming into the lacuna is moving into the core. So we'll have lesser number of free cholesterol here because they are modified and they're going to the core. We have phospholipids also, surplus phospholipids, abundant phospholipids are there in the HDL shell, because every time LCAT is acting, one phospholipid is being sacrificed, one lecithin is being sacrificed. That lysolecithin which is formed no more stays in the shell of the HDL, that rather falls to the plasma. Now this HDL3, because it's having lacuna, and of course all the apoprotein ApoA1C and D it is already having. So now this lacuna again accommodates the cholesterol from extrahepatic cell here, and that new cholesterol is again converted by LCAT action to cholesterol ester and moves into the core. So this process of LCAT action and movement of cholesterol in the form of cholesterol ester within the core of the HDL particle continues for a long time, and this converts this HDL3 particle into a bigger particle which is now called HDL2. The difference of HDL3 and HDL2 is the size, and that size difference is because of the content of the cholesterol ester in the core section. This is having more cholesterol ester in the core section because of such kind of action of LCAT enzyme, which I have just explained you some time back. Now this HDL2 is having more of the cholesterol ester in the core section. More lipid, less density, less lipid, more density. That is the fundamental fact which you should know whenever you discuss the lipoprotein, whenever you learn the lipoprotein metabolism. So now HDL2 is having more of the lipid, so its density is less. HDL3 is having comparatively lesser amount of lipid, so its density is more. Right, so this is how you should know that there is a difference in the density, in the size, in the content of lipid in various forms of HDL. Less the lipid, more the density, more the lipid, less the density it is. Now if I talk to you about HDL2, this also is having the phospholipid in the shell, and of course the lacuna, because whatever cholesterol is coming here, it's modified to cholesterol ester and moving to the core section. And in addition to this lacuna, the shell of the HDL is having the phospholipid, the free cholesterol here and there is scattered like this. Now this lacuna also accepts the cholesterol via those transporters, and that cholesterol again is modified to cholesterol ester and moves into the core. So this HDL2 continues the process of reverse cholesterol transport. It accepts the cholesterol from extrahepatic tissue with the help of LCAT. It converts it to cholesterol. It continues. This action of LCAT continues even on HDL2. But one thing you should know, this LCAT enzyme is under product inhibition. So when more and more amount of cholesterol ester is accumulated, when the level of cholesterol ester within the core of HDL2 particle exceeds the limit, and that point of time, the excess cholesterol ester starts imparting the product inhibition on LCAT action, and suppose it is having more and more of the cholesterol ester which is imparting product inhibition on LCAT action, LCAT will no more act on this HDL2 particle. So that time the last incoming cholesterol will seal the lacuna, will plug the lacuna, and the shell will be complete in itself. This Last cholesterol will not be modified to cholesterol ester because of product inhibition of LCAT enzyme, and we call it as saturated HDL2 particle, which is no more in a situation to accept the cholesterol from extrahepatic tissues. So what you have learned till now, that HDL came in a flattened structure form, which was called discoidal HDL, and slowly it started taking shape, a spherical shape. It became HDL3, then it became HDL2. And I said that at one moment when a lot of cholesterol ester is accumulated in the core section, the HDL2 is no more in a situation to accept more cholesterol from extrahepatic tissue. This time this HDL2 moves to the liver, where it is metabolized. Now let's see what is the destiny of this HDL2 at the liver. Suppose this is liver and this is HDL2, which is having A1, C, E, lot of cholesterol ester in the core, phospholipid and free cholesterol in the shell. There are two different fates of HDL2, at the hepatocyte. Which fate the HDL2 will be meeting depends upon the rate of flow of the blood. In fast circulation, what happens? The HDL2 is converted to HDL3 and is sent back to the plasma. This is due to removal of some of the cholesterol ester, and the tag which the HDL2 got in exchange with the remnant particles, which I'll be dealing little later. That tag and cholesterol ester is taken up by the liver. Not all cholesterol ester, some cholesterol ester, but all tag is taken up by liver. It's metabolized by liver, and leftover particle of HDL is sent in the form of HDL3, which is still having ApoA1, C, E, some cholesterol ester in the core, phospholipid and free cholesterol in the shell. Now this HDL3, which has come out from the liver into the plasma, enters the reverse cholesterol transportation, and further it is accepting cholesterol from extrahepatic tissue, and by the mechanism which I described above will be converted to HDL2. So this is called HDL cycle, where HDL2 is reaching the liver and is converted to HDL3, which is coming to the circulation. Again, it is working and accepting the cholesterol from extrahepatic tissue. See, when the cholesterol ester is taken up by liver, the product inhibition on LCAT is abolished. So the LCAT starts acting once again on this HDL3 and allows the lacuna to be formed, and that lacuna accepts the cholesterol from extrahepatic tissue. So this is number one fate, which is seen when there is rapid circulation. In sluggish circulation, what happens? The HDL2 is more intensively getting metabolized by the liver, where each and every component is taken care by the liver. It's getting metabolized except A1. Now the cholesterol ester will be taken up. The tag which I said was this HDL2 was getting in exchange during circulation from the remnant particles, which I'll be dealing shortly, also will be taken up. So tag is taken up, cholesterol ester is taken up by hepatocyte, ApoC and ApoE are disintegrated, phospholipids are metabolized, cholesterol ester are all taken up. And this kind of HDL2, where each and every component of it is metabolized except ApoA1, that ApoA1 escapes to the plasma, where either it is filtered out in the urine and lost from the body, or it is getting embedded in a missile-like structure, the missile-like structure, which are very rarely found in the plasma. This is missile, which is made up of phospholipid and free cholesterol. So it's very rare to see such kind of missiles which are produced due to cholesterol and phospholipid sonication in the plasma. This kind of structure is called missile, and in this ApoA1, if I take black color for the ApoA1, this ApoA1 is embedded in this kind of particle, which is now formed into the plasma, is called pre-beta HDL. This is very rare to be formed. So the escaping ApoE1 rarely is going and helping in formation of pre-beta HDL. The core of pre-beta HDL is totally empty. Majority of ApoE1 is filtered out in the urine and coming out from the body, and a very small amount of ApoE1 is embedded in pre-beta HDL. This pre-beta HDL is very active form of HDL. It's in fact most active form of HDL because it's having scavenging capacity, because it's having A1. Because of this A1, the peripheral cholesterol is converted to cholesterol ester, and that's moved into the core, creating a lacuna here. So again, the lacuna is created by LCAT action. See, LCAT can very well act on pre-beta HDL because it's activated by A1. It's A1. So once LCAT is acting, you know what is happening. The lacuna is created. Now this lacuna accommodates cholesterol from extrahepatic tissue. And that cholesterol, when accommodated here, is again modified to cholesterol ester by next cycle of LCAT action, and that cholesterol ester again moves into the core. So again more cholesterol comes, and that's converted to cholesterol ester and moves into the core. So slowly pre-beta HDL starts taking a discoidal shape like this, where some of the cholesterol ester is there in the core section having ApoE1. C and E initially is not there on this pre-beta HDL, yeah, but if donated by mature HDL, though it can have C and E, it will have phospholipid, it will have lacuna, because whatever cholesterol is coming in this lacuna is going into the core section. This kind of structure, what you see in this picture, is nothing but the discoidal HDL. So if you see, the discoidal HDL is not only coming from liver and GIT, it is also originating in the plasma from pre-beta HDL when LCAT is acting on that pre-beta HDL. Now this discoidal HDL will join the reverse cholesterol transport process, and the method which I have explained you just now, again that discoidal HDL is converted to HDL3, then HDL2, and that HDL2 is meeting the liver, and there its fate is decided. So this is how the whole of the HDL metabolism, you need to learn that starting point is the formation of discoidal HDL from the liver and GIT, and then how it takes the spherical shape, and then when it reaches the liver, how two different fates of HDL is there at the liver surface. One is the conversion of 2 to 3, another is conversion of HDL2 to ApoE1. Then seeing the fate of ApoE1, which I have told you, rarely it becomes part of pre-beta HDL. It's a very tiny HDL, very rare to be seen in the plasma. And this becomes discoidal HDL after LCAT action, and discoidal HDL becomes HDL3. When some more cholesterol is modified to cholesterol ester and is packed in the core, then it becomes HDL2. So we see practically there is no loss of HDL. The system is trying to conserve the HDL. Liver is always trying to conserve the HDL, either by converting it to 3 form and resending it to the circulation, or converting the HDL2 to A1 in the hope that it becomes part of pre-beta HDL and gets involved in the process of reverse cholesterol transport once again. The transporter, the extrahepatic cell transporter, which I said that they are responsible for fetching the cholesterol from extrahepatic cell to the HDL particle. There are so many types of transporters, which you call them as ABC, A1, ABC, G1 transporter, ABC, A1, G1, B1, ABC, B1 transporter. So ABC is ATP binding cassette, A1, G1, B1 transporter. Then in addition, you have scavenger receptor B1. These are various transporters which are present at extrahepatic tissue, which fetch the cholesterol from extrahepatic cell to the HDL particle. This SRB1 is not only present at extrahepatic tissue, it is also found at the liver. When the cholesterol ester need to be delivered to liver, that time SRB1 play a role. So SRB1 is found on hepatic as well as extrahepatic cell, and it has got dual role in the HDL metabolism, because at the liver you can see liver surface from lipoprotein, it is delivering the predominant cholesterol ester to the hepatic tissues. And extrahepatic tissues, if you focus on the role of extrahepatic tissue, SRB1 role on extrahepatic tissue, What it is doing, it is transferring the free cholesterol to lipoprotein from the tissue to the lipoprotein. So at the extrahepatic level, from the tissue to lipoprotein, and that is free cholesterol, and at the liver surface from the lipoprotein to the tissue, and that to cholesterol ester. So this SRB1 is having dual role in the HDL metabolism. During this circulation, when HDL2 is, I said that there is some triacylglycerol which is accommodated in its core. Let's see from where it is coming to HDL2 core. So for that, I would like to draw this separate diagram. This is HDL2, which is having lot of cholesterol ester, and the LCAT has stopped acting on it. It has got A1, it has got C, it has got E, it has got phospholipid and the cholesterol in the shell, but it is no more able to take the cholesterol from the extrahepatic tissue because of no action of LCAT, because of product inhibition of LCAT by excessive cholesterol ester in the core. So when this HDL2 is going to liver on the way, at times it is finding certain remnant particles, and these remnant particles are chylomicron or VLDL remnant particles. These remnant particles are small lipoprotein particles which are end products of VLDL and LDL, VLDL and chylomicron metabolism. They are called chylomicron remnant and VLDL remnant, otherwise called IDL. They are having some triacylglycerol in their core. In addition to some cholesterol ester, they have some triacylglycerol also in their core. So with the help of one protein that's called cholesterol ester transfer protein, some of the cholesterol ester is transferred to these remnant particles in exchange for triacylglycerol. So this is called exchange of lipid from core to core. So HDL2 donates cholesterol ester, and triacylglycerol is taken up from the remnant particle. This kind of exchange increases the capacity of HDL2 in the plasma. It can act for some more time because some cholesterol ester is removed from its core. So LCAT becomes functional once again on this HDL2 particle because of removal of cholesterol ester, which was imparting the product inhibition on LCAT. When that product cholesterol ester is removed from HDL2 core, that time LCAT starts acting once again on HDL2 and starts creating a lacuna which can fetch some more cholesterol from extrahepatic tissue. So this kind of exchange facilitates this HDL2 to work for some more time in the plasma until it is again getting saturated with a lot of cholesterol ester and LCAT stops acting once again. So this kind of exchange delivers the triacylglycerol in the HDL2 core, the tag which I was talking to you about at the hepatic cell, where it is being delivered to hepatic cell, and that triacylglycerol, that peripheral phospholipids, they all are metabolized by hepatic lipase enzyme. Hepatic lipase enzyme. So thank you very much.